All right, this lesson is about DNA structure. Okay, DNA is also known as deoxyribonucleic acid. Have a practice, try and say it. Deoxyribonucleic acid, and they all refer to little bits and pieces. We understand chemical names have very important parts to them, and the deoxy part is related to the deoxyribo sugar that is involved in DNA, and we'll later look at RNA, which has a ribose sugar. So let's find the difference, spot the difference, spot the difference. Here's our difference here. All right, the nucleic acid part is really important because if you think back to late last year, we did our 4 kinds of organic macromolecules, and nucleic acids are one of them. And nucleotides are the building blocks that make up the nucleic acids. And we're going to come and talk about amino acids a little bit later because they are the building blocks of proteins. But really what we're looking at is one gigantic molecule, and it's made up of nucleotides. Now, DNA versus RNA we'll look at a bit later on, but we need to know that there's a structural difference. We've got a single strand here versus a double strand DNA, and we also have differences in the nucleotides that make it up. As I mentioned before, we've got the ribose sugar in the RNA, making it RNA, and the deoxyribose in the DNA. Okay, you're going to see this, the DNA is going to be shown in so many different ways, and it doesn't really matter which way you draw it or illustrate it in any other way that makes sense to you, but you're going to see it in so many different forms. So we've got this twisted ladder kind of structure here, we've got the straightened out ladder version, and you've got a more chemically horrifying looking one, but really important. This will come into play in a moment. So sometimes they straighten it out and do it in a nice kind of Lego looking manner. Sometimes it's a bit more cartoony. Sometimes we can actually see more structure like we have up here about the type of sugar we've got, so the S for sugar. And sometimes we see that it's not just a simple twisted ladder structure all the time. There's actually more complex shapes to it. We're not going to have to worry about that, but just understand that it is not as simple as we think it is. We're showing you a very simplistic model of this. All right, we're going to kind of focus on these ones. This is important, but it doesn't show enough detail. This is nice straightened out. It's showing a little bit more detail with the 3' and the 5', but it's not everything we need. This is almost too much, so we're kind of going to work in the middle here with this one. All right, DNA is a double helix. It is a twisted ladder. You will see versions of it in your lab, and you will see different types. We've got a chemical version with all the little atoms, which is huge, and we have a very simplistic model as well. It has a sugar and phosphate backbone as the uprights. I'm going to call that the uprights of the ladder. And you can see this nice little pentagon here is our sugar. Our green part is our phosphate, and you can see that the nucleotides are actually bonded there to the sugar. But there are nitrogenous nucleic acid bases, right? Nucleic acid bases. And really what we're looking at here is their main component is nitrogen, and these are the rungs. It's really important to understand that there are 4 rungs in DNA, 4 of those nucleotides: adenine, thymine, guanine, cytosine. The INE on the end is indicating that there is nitrogen involved. There is another base that we'll learn about with RNA, but we don't need to know that right now. It's really important to see that there are matches here. A or adenine will always match with T, thymine, always. There is no exceptions, except in RNA. I'll come back to that. You can see here this has got a bond, and it's a double bond. Cytosine and guanine match together as well. So I like to think of it as the letters made out of straight lines match together, and the letters made out of the curly lines match together. Very simple. So as I said, there's a weak hydrogen bond joining the complementary bases, and that's how we will refer to them. They are complementary bases. And you can see the C and G have a tighter bond going on there. We talk about the human genome, and the human genome is simply all the letters written out to figure out, hey, across our 46 chromosomes in our cell, what are they, right? And obviously we're going to have variations amongst people, but you can see that, there's a big repeat here. Maybe I have 4 of them, you have 3 of them and a T. I don't know. And how much impact that will have on our genome and what we look like is going to be very different. It's not just a really simple ladder. it's called an anti-parallel ladder. Okay, that double helix is anti-parallel, which means even though these things run parallel, they run in opposite directions. Now, if we go back and have a look at the picture, it doesn't look like there is a direction in this particular model, but really they do run in a particular way, and it's all about the sugar molecule. So there's a particular way in which these are twisted up as well, and they talk about the minor groove and the major groove. So just the bit where where the nucleotides would be versus where there isn't any. And the important part of 3', 5' is that the ends relate to what carbon the sugar is attaching to the phosphate here. So at this end, we've got the 5' end is carbon number 5, whereas this one, it's the 3'. So you're going to keep seeing that on different models. Just understand that they are going to go in different directions. If it's 5' here, then the opposite or the complementary strand is going to be 3' and vice versa down here. So this is really only looking at one side of the ladder. So here it is all put together. Our 3' is opposite our 5', and you can see that it is structured like that. The sugars are all pointing this way, but on the opposite side, the sugars are pointing in a different direction. Right, enough chemistry. We have to talk about going from the really, really small to the really, really big. And what this giant piece of DNA is doing is coding. It's a big instruction kit. It's instructions for proteins. Right? And the only way we can do that is by making those tiny little amino acids that make up the chain that is the protein, and we have to code for it, right? This is the instruction book or the recipe book, whatever it is. And on a big piece of DNA, we have important pieces. We have exons and we have introns, and they mean different things and they do different roles. Exons are the regions containing instructions for coding that protein, right? And the introns are kind of the bits in the middle. I wouldn't say they're junk. You sometimes hear that they're junk nucleotides, that they don't mean anything, but they are in there for a particular reason. Sometimes they are repeats of bases, so A for a really long time, and certain chromosomes have really long chunks of those repeated bases, but they are called introns because they are intragenic regions. They are just within the gene. A gene is then, therefore, it might be this whole thing. This whole gene might code for how long your nose is. I have no idea. But both the introns and the exons make up this particular section, okay? Now, down the track, our body goes, we don't need these bits. We don't need the introns. They aren't useful to us. So we're going to snip them out. I'm going to condense all the good stuff and stick it together. And from that set of instructions, we can make it. So think of it like an encyclopedia where you only need every second entry to do your assignment. So eventually, even though you photocopy the whole thing, you're going to start pulling out some of those things and using the bits you need. DNA is massive, like massive. I'll actually get you to look up how big it is, but we're talking every single chromosome is massive, and then you've got 46 of them. And they have to condense down into the nuclei of every single one of your cells, except for the red blood cells and the gametes that are in your body. So the way it does that is not only that it's this ladder, but it starts to twirl and twirl and twirl and twist and twist and twist, and starts to fold itself around these histone proteins. Now they'll come in little clusters, and then once that happens, it starts to fold in a more 3D way as well, and it starts to coil and coil and coil until it condenses down to the point where we can see it. Now at this stage, we can't see that. I can't get you to extract DNA from something, put it under the microscope and see the ladder. It would be nice, but we can't. But we can see it at this point where it's condensed right down because it's basically folded in on itself over and over. That is the kind of thing we're talking. We're going from this ladder to folding it around those histones to folding it around itself again and again and again and again. Now, there's a massive error in this picture, and I don't like it because this is called chromatin. There should not be an E on the end of there. It is simply chromatin, right? This is chromatin, these are chromatin loops, and then this is condensed chromatin before it becomes that full chromosome. So sometimes I use DNA and chromosome interchangeably, and I need you to understand it's really saying the same thing, but the chromosome contains all of those histone proteins, and then it's folded around on itself over and over. Your genetic materials can be in your chromosome or it can be in your DNA because your chromosome is made of DNA. We also have to talk about RNA. Now, RNA will come into it down the track when we learn about how we go from those instructions to the protein, but we need to know some main differences. DNA has got that double strand, and it's got the T, the A, the C, and the G, but over here the RNA is only single-stranded. Now, some viruses have single-stranded a single-stranded genome, right? We know they aren't alive, but they do run off RNA. RNA plays a very different role in our body. And you'll have a quick note that there is no thymine, right? So A, or adenine, actually matches up with uracil down the track. Now, if that's what we're looking at, which is single-stranded, then being complementary doesn't matter, but it will later on. And what the roles that we're playing here is like, this is the instruction book. The DNA in your cells is the instruction book saying, hey, you can come and photocopy me, but you can't take me anywhere. It has to stay in the nucleus. So the DNA stays in the nucleus, and the RNA can make a complementary strand, which in turn will become that protein. But this is the traveler. This one stays exactly where it needs to stay, in the nucleus.